Picture this. Deep inside your heart, a myocardial cell is just doing its thing. It's working perfectly, beating away, keeping you alive. But then, suddenly, its oxygen supply gets completely cut off. What happens next is a high-stakes, microscopic fight for survival. Within literally just a few minutes, a massive domino effect begins. Since that cell is now starved of oxygen, its internal power grid just completely shuts down. ATP, which is basically the cell's energy currency, plummets. And without that ATP, the crucial sodium-potassium pumps on the cell's surface just fail. So sodium rushes into the cell. And because biology absolutely loves balance, water follows that sodium right on in. The physical consequences here are rapid and honestly pretty dramatic. The cell actually begins to physically bloat and swell up with all that excess water. So looking at this bloated, struggling cell, you gotta ask the big question. Is it dead? Well, actually no, not quite yet. If that oxygen blockade is cleared early enough, this injured cell can entirely recover. It's resilient. But if the injury continues, this damage crosses a fatal threshold and becomes completely irreversible, which absolutely leads to cell death. Okay, let's dive into this explainer and decode this microscopic thriller. We're going to cover the stressed cell, reversible cell injury, the point of no return, necrosis versus apoptosis, and finally, some classic pathology examples.
First up, Section 1, the stressed cell.
Normally, a healthy cell perfectly maintains its internal balance, a state we call homeostasis. But when it's bombarded by stress, it's forced to adapt. Think of a timeline. At 0 minutes, we've got normal homeostasis. About 5 minutes in, the cell is really trying to adapt to the stress. But hit 15 minutes of severe stress, like total oxygen loss, and we enter the zone of reversible injury. Now drag that out to 60 minutes or more, and that persistent insult violently pushes the cell right off a cliff into irreversible injury and death. Moving right along to Section 2, reversible cell injury. As long as its foundational structures stay intact, the cell can completely bounce back if you remove whatever is damaging it. Here's exactly how that first physical sign of injury, cellular swelling, plays out during ischemia, which is just a drop in blood flow. Step 1, blood flow and oxygen drop. Step 2, oxidative phosphorylation decreases, so ATP production falls. Step 3, those ATP-dependent ion pumps fail. Step 4, sodium builds up inside, pulling water along with it. And step 5, the cell swells up. This swelling is literally one of the absolute earliest physical signs that a cell is in distress. But hey, swelling isn't the only red flag. The second classic reversible change is fatty change, or steatosis. Instead of water, we start to see lipid droplets, specifically triglycerides, accumulating right inside the cytoplasm. It's almost like the cell is frantically hoarding fat for a long winter. We see this classically in the liver, and it's often driven by toxins like alcohol, hypoxia, or metabolic dysfunction. But just like swelling, if you get rid of the toxin or fix the metabolism, those liver cells can clear out the fat and totally survive. Now for Section 3, the point of no return. So what happens if the damaging stimulus just doesn't stop? If that blockage stays put, the cell crosses a terrifying line. Irreversible injury is exactly what it sounds like. Recovery is now entirely impossible. Fundamentally, this point of no return is defined by two massive structural failures. First, you get severe mitochondrial dysfunction. This means the cell's engines are permanently destroyed and energy failure is absolute. And second, profound membrane damage. Membrane integrity is absolutely crucial for survival. Once that plasma membrane is severely damaged, stuff from the outside floods in, and the cell's vital internal contents just leak right out. And this brilliantly illustrates what happens when those walls break down and ion gradients collapse. Massive amounts of calcium just flood into the cytoplasm. Calcium acts as this ultimate cellular destruction activator. This sudden calcium surge turns on a frankly terrifying squad of destructive enzymes. Phospholipases start shredding the cell's membranes, proteases tear apart vital proteins, endonucleases chop up the cell's DNA, and ATPases burn through whatever precious little ATP is even left. The cell is quite literally being dismantled from the inside out. As if that wasn't enough, this internal dismantling gets compounded by another major threat, oxidative stress. Those damaged mitochondria start churning out excessive reactive oxygen species, or ROS. These unstable molecules just wreak havoc everywhere. To remember exactly what they destroy, I love this high-yield mnemonic. ROS leads to LPD. L is for lipid peroxidation, which destroys membranes. P is for protein oxidation, causing major dysfunction, and D is for DNA damage, which causes lethal mutations. It's essentially a triple threat to the cell's survival.
Which brings us to Section 4, necrosis versus apoptosis.
Once irreversible damage is done, the cell is going to die. But how it dies matters immensely. Honestly, if you take away only one major thing from this explainer, let it be this distinction. Necrosis is incredibly messy. The cell swells, the plasma membrane completely ruptures, and all those internal contents and digestive enzymes spill out into the surrounding tissue. That triggers a massive, angry, inflammatory response. Apoptosis, on the other hand, is highly regulated. The cell neatly shrinks, fragments into these tidy little packages called apoptotic bodies, and is quietly consumed by phagocytic cells. All of this happens with minimal to zero inflammation.
Let's zoom in on that messy explosion of necrosis for a second. As the cell undergoes enzymatic digestion, its nucleus goes through a very specific three-step sequence of absolute destruction. It's super easy to remember as P to K to K. First is pycnosis, where the nucleus condenses into a tight, dark, shriveled little mass. Next is karyorrhexis, where that condensed nucleus basically shatters and fragments into tiny pieces. And finally, karyolysis, where those nuclear fragments completely dissolve and vanish. In stark contrast, apoptosis is controlled cell deletion. It's a programmed suicide, and it's triggered by two main pathways. The intrinsic pathway kicks off from internal cellular stress or DNA damage. That causes the mitochondria to release factors that activate an enzyme called caspase-9. Then you have the extrinsic pathway, which is triggered by outside signals hitting death receptors on the cell surface, and that activates caspase-8. Interestingly enough, both of these pathways ultimately converge. They both activate executioner caspases, like caspase-3, which then neatly dismantle the cell from within, packing it all up for safe disposal.
Finally, Section 5, classic pathology examples. So why does all this microscopic biology actually matter to your health? Well, it's because we can categorize necrosis into patterns that perfectly match up with real-world diseases. When ischemia hits the heart, causing a myocardial infarction, we see coagulative necrosis. That's where the architecture of the dead tissue is remarkably preserved for a few days. But if an infarction happens in the brain, enzymatic digestion takes over super rapidly, causing the tissue to literally turn to liquid. We call that liquefactive necrosis. Tuberculosis in the lungs forms a cheesy-looking granuloma, known as caseous necrosis. Acute pancreatitis releases lipases that destroy surrounding tissue, causing fat necrosis. And severe immune reactions in blood vessels cause fibrinoid necrosis. So you see, the specific type of cell death directly defines the disease.
We've journeyed all the way from a healthy cell, down through the timeline of stress, past the point of no return, into the messy reality of necrosis and the neat packaging of apoptosis. But I want to leave you with this final, somewhat wild thought. Right at this very moment, your body is constantly facing environmental stress. So how many of your own cells are silently negotiating this microscopic line between adaptation, reversible injury, and apoptosis? Right now, literally as you listen to this. It's a continuous, high-stakes battle happening inside you every single second. Thanks for joining me on this explainer, and keep questioning the incredible machinery that keeps you alive.